Hello, welcome to the latest episode of Data Unchained. I'm your host, Molly Presley. Before we jump in and introduce today's guest, I'd like to tell you a little bit about what this podcast is focused on. Essentially, we're talking about how the paradigm for data access has changed. In today's decentralized world, getting data to remote workers, distributed applications, moving it around between data centers and cloud regions can be a real challenge. Data Unchained digs into both the challenges as well as the solutions that make data an asset as a global resource. Today's guest is Kevin Libuit. He's the CTO of Theiagen. Kevin, thank you for joining us.
Thank you, Molly, for the invite and having me on this show. It's been really cool kind of digging into the podcast history, seeing the different wide range of guests you've had to speak on different topics, some of which is really specific to my world of genomics and bioinformatics, other of which cover things that are like really big ideas. Like I caught that episode with Roxanne, and she had just ideas and experience, like just unbelievable depth and breadth. So it's definitely a privilege to be on this and to be kind of that part of that cohort of interviewees on Data Unchained here.
Awesome. Thanks. I don't know if you saw, probably the funnest one was when we had Adam Savage on because he was making a sword in the background.
I saw that.
That was interesting. And then you have Roxanne talking about big global trends that affect humanity. It is fun to have the breadth of conversation.
You certainly have a cool job in being able to kind of see the gamut of subjects that are all just in some way related to this conversation of data and access, and it's just wild to see how it kind of proliferates so many different industries. So as I said, very, very cool to be a part of this.
Very cool. So tell me, before we talk about the company you work for, would you tell us a little bit about Kevin? Kind of how did you get to the role you're in today as CTO of a genomics company?
Yeah, so my career started in pathogen genomics when I was studying at James Madison University over there in Virginia. And I was in a traditional microbiology laboratory where really our focus as a research group was looking at antimicrobial resistance and how sort of agricultural practices introduce these antibiotics into the ecosystem and tracking the way in which it's impacting microbial communities. And when I first started in the sciences, I was a bench scientist. I had a lab coat, I was working with bacteria with my hands directly, and I was looking to see how resistance was being expressed by these microorganisms. And as we were looking deeper at this question, the sort of natural next question was, what's the genomic content behind these antimicrobial resistance that we were seeing? And so this was my first experience looking at DNA sequencing and the bioinformatics to make sense of these data. So this was around 2014, 2015 when I got my master's degree and was first introduced to these concepts of microbial pathogen genomics and bioinformatics in general.
Yeah. So in your work, has it been primarily lab related, or did you make a shift over to technology and computers at some point in that path?
Oh, that's a great question. So I started at what we call the wet lab. When I was actually running the experiments, I was in the laboratory growing up these bacteria. But then by nature of the questions we were asking, we were generating so much data, I had to learn the dry lab techniques. I had to learn a little bit of bioinformatics, a little bit about programming, so that once we were able to generate these data, I had to find a means by which we can actually make sense of these data. And actually that right there was my shift and kind of became the sort of microcosm of my career in working with so many different laboratories who have built up those wet lab capabilities to generate these data. They all have the same problem that I had. How do I now implement the bioinformatics to make sense of these data?
Makes sense. So just maybe for those who are not real familiar with the life sciences space, what is bioinformatics?
Oh, yeah, great. So bioinformatics is really sort of this mix of a field in that we take a lot of the principles and approaches from data science and computer science and apply those approaches and techniques to make sense of large biological data sets. I think there's maybe a number of different ways in which people define bioinformatics, but I think that kind of captures it well. We employ data science and computer science techniques to try to elucidate some insights from biological data. In my background specifically, and what we do at Theiagen is we're looking at big data sets of pathogen genomic data. So DNA coming from infectious diseases like SARS-CoV-2, E. coli, Salmonella, tuberculosis, etc.
Okay. And so in that space, certainly, I think everyone's heard of most of those diseases and pathogens. What is it that you are looking to solve at Theiagen? What is the, kind of passion of your country, your company, and kind of are you coming up with drugs? Are you understanding how they reproduce? What is it that you guys are looking to uncover?
Yeah, that's a great question. What we're trying to do is answer that same question I was trying to ask when I was at my laboratory in JMU. Is there are laboratories that are generating these data? How do they get the resources and infrastructure to make sense of these data in terms of implementing the bioinformatics? So to kind of even go back to the narrative route I was starting on in terms of my career, right after I earned my master's degree at James Madison, kind of serendipitously, it was the same time where the whole U.S. public health infrastructure was turning to sequencing and bioinformatics for their pathogen surveillance network across the U.S. So 2016 is when I joined the Virginia State Public Health Laboratory. And they were one of the first state public health laboratories to begin sequencing and performing bioinformatics for looking at foodborne pathogens that are kind of being disseminated or spreading across the state, and trying to look at how genomic data can be used to better understand how it's spreading and why it's spreading, so that it can eventually inform action in terms of, how do you stop the spread of these infectious diseases. And as I said, this was when the whole U.S. infrastructure was starting to make that transition. So I was really fortunate enough to be one of the first bioinformatics scientists at the state level. In fact, I was one of only 4 in the U.S. State system. There was myself, Joel Savinsky, who was at the Colorado State Public Health Laboratory, Sean Wang over there in Minnesota, and Kelly Okeson in Utah. And we had really specific challenges of bioinformatics implementation. There was not only the analytical capabilities in terms of lack of software to properly analyze these data, there was also the conversation of the appropriate compute infrastructure itself. A lot of laboratorians, they had maybe their desktop computers and were looking at assays that they can kind of put into an Excel spreadsheet and almost do their data science on a couple of Excel sheets. Whereas, we were generating gigs of data, sometimes terabytes of data, and we needed the appropriate infrastructure to run the software even to make sense of these things. And then beyond that was training the public health community. It's really a whole new language, it's a whole new lexicon of looking at these types of data, these types of output, and making sense of these types of reports to properly inform what was going on. So in my career, I had that kind of front row seat at the Virginia State Public Health Lab of understanding what are the challenges, hurdles, and even solutions that allowed public health laboratories to kind of overcome these barriers. And in the beginning was really working with those 4 scientists specifically to create a kind of community practice. We formed the State Public Health Bioinformatics Working Group, or StaPH B, that really, over my time in Virginia, snowballed into a whole nationwide community where we had practitioners from every state public health laboratory trying to share these ideas of how do we get the analytical tools, how do we get the infrastructure, and then how do we train the scientists to actually make sense of these data. And so I was at the Virginia State Public Health Lab until about 2020. And I think it was actually 2018, 2019, Joel Savinsky, who I mentioned was at the Colorado State Public Health Lab, his ambitions as a practitioner sort of superseded his role at the state. So this is actually when he stepped out of his position in Colorado and started Theiagen Genomics. And he started working with states directly beyond just the Colorado State Public Health Lab to take a lot of those lessons we were learning and try to expedite their implementation as more of a sort of public-private partnership. And so I saw him kind of step out and make his move and start Theiagen. And then in 2020, I left the Virginia State Public Health Laboratory and joined Joel. And in the beginning, it was just trying to figure out, with a lot of laboratories, we knew the utility of things like pathogen genomic data. As I said, looking at things like foodborne infections and different hospital or healthcare associated infection control. And our small community understood the utility of pathogen genomics, and we were, very convinced that this is going to be the required technology to perform these kinds of, or to support these types of investigations. And then we thought, maybe we had a 10, 20-year layout of how this technology gets implemented across, different states and different countries around the world. But then in the face of COVID-19 response efforts, it became the only technology, it became the premier technology for laboratories to really understand what was going on. So what we understood as a small community in terms of its utility and how it can transform public health, COVID-19, so much more funding got put into the space to expedite that adoption. So every U.S. public health laboratory quickly gained those capabilities to generate these data for SARS-CoV-2 sequencing. And then just this half step behind that, they needed the same capabilities to perform the bioinformatics and make sense of these data. So that's really where a lot of our work in Theiagen started. So 2020, 2021, or at least my work at Theiagen started. 2020 is when I joined, and it was Joel and I working with laboratories to set these bioinformatics systems up. And now, 3 years later, we're a small team of 16 scientists all over the world working with public health laboratories through the global community, answering these same questions, not only for SARS-CoV-2, but also other infectious diseases of public health concern.
So I think that the way data is shared has changed a lot. There's a lot of regulations, of course, around it.
Yes.
And then there's technology hurdles as well. But when you talk about sharing information across states with different groups, where are the obstacles? Is it a technology problem of moving data from one place to another, or is it more policy level?
Yes, and both of those are very much true. And there's a huge need in ensuring a lot of these data get shared because, again, going back to sort of what the public learned through COVID-19, none of this is isolated to a specific government jurisdiction, right? Like things spread across borders. These pathogens know no borders. So we have to be able to have that global perspective of how these pathogens are spreading across jurisdictions. So there's been really huge efforts of sustaining these internationally accessible databases with some of these critical pieces of information. So like a big push or a big effort was made or is continuously being made for public health laboratories to be able to submit their data to these internationally accessible databases, things like GISAID or NCBI, where information about the sequencing data itself can be shared, but also crucially important is the metadata, the information about that sequencing data in terms of when it was collected, where it was collected, and other sort of vital information that give context to the infectious disease investigations there. So the challenge there, as I said, it's sort of a yes and, but I think the way in which a lot of laboratories are addressing that is simply through education. Speaking with IT, speaking with the data owners, and speaking with the epidemiologists and other partners to understand what data will be shared and what data won't be shared through these different forums. And it's been amazing too, having in the face of COVID-19, sort of this global community come together to understand, and communicate the critical importance of putting these data out there. So there's a lot more willingness to share these data, but again, there's still those apprehensions and challenges of the data that is allowed to be made available there.
Do you think that this will be limited to COVID-19 or, God forbid, the next pandemic? But there's many other public health issues going on. Is a lot of this infrastructure going to be used for other diseases as well?
Absolutely. And we're already seeing that because, I think the way we approached the bioinformatics development, when I say we, not only Theiagen, but really throughout the whole public health community, with that understanding that these technologies are going to be organism agnostic. And we're looking at DNA sequencing already. So there's already the capabilities. If you can sequence the DNA of SARS-CoV-2, you more or less have the capabilities to sequence the DNA or the genomes of multiple pathogens. And so in turn, we wanted to make sure that the bioinformatics solutions were also organism agnostic, or as organism agnostic as possible. So you're seeing a lot of adoption of just general infrastructure like cloud compute platforms, the use of interoperable software through like Docker containers and workflow managers to make even the bioinformatics pipelines themselves all that more distributable and accessible to different laboratories, all of which are not in any way SARS-CoV-2 specific, but the general principles there can be quickly pivoted to things like the other pathogens we talked about. In fact, we had a really close collaboration with the Massachusetts State Public Health Laboratory and the Grubal lab over there at Yale University in the face of the Mpox outbreak, where we were able to take a lot of those lessons learned from SARS-CoV-2 and from the wet lab side to the dry lab side, pivot it in the face of another emerging infectious disease. So the Yale University, the Grubal lab at Yale, they were able to develop the wet lab capabilities to sequence Mpox, and we were able to work with the Broad Institute of MIT and Harvard, as well as the Massachusetts State Public Health Laboratory, to make sure the bioinformatics capabilities that we had developed for SARS-CoV-2 could be, again, repurposed or pivoted towards Mpox sequencing and analysis.
So who pays for all of this? It's, a lot of public meets private kind of world. The infrastructure, the research, the collaboration, is that companies like your own, or is there government funding? How does that go about?
Sure. Nearly all of our work has been supported by CDC funds that make it either directly that we're working directly with the CDC, or that the funding that gets distributed to the states and then allocated to the different stakeholders to ensure that they have these kinds of capabilities and resources to perform this public health good. And so like some of the offices at the CDC, for example, the Office of Advanced Molecular Detection, currently headed by Duncan McCannell there, they've allocated a lot of funding and resources to ensure that the U.S. public health system has the sequencing capabilities as well as the bioinformatics capabilities to support these types of investigations. Because I think what we've seen over the past, maybe 10 years now is how much more powerful these data are relative to maybe traditional wet lab techniques of like molecular biology, pulse field gel electrophoresis for maybe the molecular biologists that are listening in. It's just orders of magnitude greater resolution to support these types of investigations. So that urgency to shift into technology is certainly understood and well funded, or at least, relatively speaking, there's financial support coming from the federal government there.
So what is the IT side of the data sharing? And when you look at the infrastructure you're using, is this all cloud-based? Are you hosting it in some kind of SaaS tools that have been designed for the industry?
Yeah, no, that's a fantastic question. So a lot, and it's worth saying too, the data or the resources we're making available for the community is for them to analyze their data. So we're not generating any data. The data analysis we're trying to put into the hands of the public health laboratories that we're working with. So what the infrastructure generally looks like is the laboratories are able to generate these data, and either they'll have some kind of cloud storage themselves, or rarely, but sometimes they'll have some kind of local data storage of the genomic data itself. And then the conversation is, how do you transfer it to the system where actually the data processing is happening? And so we've been able to find a couple different solutions for this. In the public health community, one platform that's been really powerful is the Terra platform that was developed and currently sustained by the Broad Institute, some great collaborators over there. And it's all cloud-based. So currently something that's hosted on GCP as well as Azure. But then the whole conversation is how do you get the data that the laboratories generated and then onto or made accessible to the Terra platform itself. And then from the Terra platform, there's this whole coordination of the actual applications or the bioinformatics pipelines that are stored in something called DocStore. So there's this repository of pipelines and workflows that we make available that's also sort of linked to the GitHub repositories that we make available. And then all that gets coordinated and orchestrated by Terra so that the laboratories themselves, they can access, they have access to the workflows, they have access to their own data to perform that analysis, all within this Terra platform itself.
Very cool. It seems that there's this trend, and certainly that's why we have this podcast, but of getting data out of a silo or an individual human's machine or lab into the public domain, or not even public domain, but into a larger access group is accelerating science everywhere, whether it's how autonomous vehicles run and gain the data off the tractor driving around shooting weeds in a farm, or, your area of focus where it's genomics, that if we can get more of the data out of the field and into analytics tools, we can make decisions so much faster. And it just seems like almost every industry, the story's the same, even though the example's a little bit different.
Yeah, that's why again it's really cool being a part of this kind of conversation with you, because I think whenever we're kind of head down doing our work, we feel as if bioinformatics and public health is so unique and the challenges are like so specific of how do I get the sequencing data from this laboratory into something like a Terra platform and then analyzing those with, the necessary workflows and things like that. But exactly as you said, there's a lot of industries that are trying to solve this exact problem. So it's helpful to see the different ways in which other industry practitioners are approaching this and the lessons learned along the way.
Absolutely. So I think something very unique to your industry is the security parameters. And, we all care about hackers and people maliciously getting access to data that shouldn't. But with all the regulations around privacy of medical data and that type of thing, I think your industry is even more regulated. How difficult is that for you to get to data sharing when you have to worry about patient privacy?
Oh, that's a huge concern. And so I should even clarify, when we're talking about a lot of cloud solutions, a lot of the materials or data that are being shared is limited to that which is not PHI. So a lot of the information, in fact, that gets uploaded to something like the Terra platform for analysis, you're really only looking at the genetic data and very low level metadata that wouldn't be necessarily classified as PII. So, again, that becomes another conversation of the data transfer is, okay, now you've got the data in Terra, maybe you've done your bioinformatics analysis and you have these reports. How then do you marry that to the sensitive data that may be housed in a different infrastructure? And so these are the conversations we're having with a lot of public health labs now is, they have maybe designated secure environments to host this more sensitive metadata, but they need still the infrastructure like cloud compute to analyze the bioinformatics or the pathogen genomics data. So these are the conversations that happen. Then how then do we take the information or reports that are being generated in Terra and then marrying that to or transferring that or marrying that with the data that is a bit more sensitive there. So those are very, very active conversations. And, I think a lot of the technologies there, the solutions are there. But again, kind of getting back to what I was saying about even data sharing in general, a lot of this is education and letting laboratories know what data is being shared, where, and how it can be ensure its data security and integrity throughout the way. And there's a lot of very smart people working with the governments all over to try to answer exactly this problem. And we feel fortunate to be able to be a part of that conversation because that's what makes it really actionable, is not just looking at the bioinformatics analysis for sort of an academic exercise, but how then do you tie that with really the patient information, the people who are actually being impacted by this, so that the epidemiologists and departments of health can inform their action more readily to hopefully stop the spread of these kinds of diseases.
The mission's great. We're talking a lot about some of the process. When you touch on metadata, you know, again, is a thing across many industries. I'm curious, how do you create metadata in your space? Like the movie and entertainment industry 10 years ago had people sitting and watching films and saying, oh, there's Brad Pitt at minute 54, and he's, in a car that crashed in minute 56, and they would manually create metadata. And they've moved to automation of how to do that with AI engines. How does it work in the genomics field when you're thinking about low level versus high level and what's coming off of a sequencer? I think it's interesting to know how that happens.
Yeah, so a lot of that is generated and taken into account and collected by the departments of health themselves. So there's a lot of epidemiologists that are in the field contacting different hospital systems or even patients directly to collect these metadata about the samples. And this is where you get the most valuable insight into each individual sample. So there's still very much a manual component of epidemiologists out there doing the contact tracing and gaining and then curating a lot of these metadata. And so those become like some of the most important information. There's certainly a shortage maybe of epidemiologists who are able to do this really detail-oriented work of collecting these metadata for each of these samples. And there's a lot of efforts to understand how to direct epidemiology efforts, right? So that's where a lot of the bioinformatics works come into play. Can we in some way identify maybe the putative outbreaks or the plausible outbreaks that can kind of narrow the scope for the epidemiologists who are then going to the field to collect more information or epidemiological information about these samples and about the patients themselves.
Interesting. Very cool. So, when we look at what's next, what are you working on now? How do you see 23 kind of unfolding and the next couple of years?
Yeah, so there's a couple of different things, right? Over the past 3 years, it's been a huge focus on public health laboratories because that's, as I said throughout my career background, that's where I came from. And that's where I understand the problems most intimately, is working with a lot of public health laboratories to set up these kinds of infrastructure. I know the laboratories are generating these data, and we kind of understand the steps it takes to get them the solutions to make sense of these data. And then as you're seeing public health kind of grow in scope, it's then kind of rubbing elbows with different but related industries, in that a lot of public health laboratories are now also working with clinical providers and maybe even the food safety side. So for us, that's kind of where we're moving now too, is working with the other stakeholders of these same types of data, looking at how on the clinical side of things, how can these reports be made most useful for the practitioners in terms of their diagnostic or medical decision making. And then also on the food safety side, this whole conversation of outbreak investigations and infection control. This is obviously of paramount importance for the food manufacturing industry as well. So that's what we're looking at, is now very clearly an aim in public health, and we came from that field, and this is kind of where a lot of our focus is. The pathogen genomics in public health is impacting a lot of those other industries well, in terms of the clinical space and food safety. So these are where a lot of our conversations and focus is turning towards.
Cool. So I think for a little bit of a call to action for those listening who'd like to learn more, and also I think it's something to be proud of. I know you were just recently published and talking about microbial genomics, and maybe talk a little bit about that paper, and we'll add a link to it for the show notes here for anybody who wants to check it out. I'm actually have it up in front of me right now. But maybe just share a little bit about that. I think COVID is near and dear to all of us, and we're familiar with it, and it's super cool to speak to somebody who's been such a part of this.
Yeah, certainly. So the paper you're talking about there, this is something that we just recently published in Microbial Genomics, and it encompasses a lot of what I spoke about in terms of accelerating the implementation of bioinformatics in the public health space. And we really talk about how this Terra platform that I mentioned helps to coordinate, or it helps to meet a lot of the needs that are specific to public health, in terms of getting laboratories access to these open source software where they have scalable compute resources and enable some sort of standardized analysis throughout the field. And in this, we highlight how just in short time, maybe 2.5 years or so, the resources we made available have been implemented in over 90 public health laboratories around the world, with millions of analyses that have taken place for their SARS-CoV-2 pathogens there. So in terms of call to action for the listeners who might be interested in learning more about this, definitely link them to my paper there. There's also other incredible groups that we've had the opportunity to work with, including the Public Health Alliance for Genomic Epidemiology, something called PHAGE. And I've also mentioned things like StaPH B, the State Public Health Bioinformatics Working Group, all of whom are doing great work to really define the critical challenges and then highlight the solutions to bioinformatics implementation in public health. I think those are great places to start to be made aware of where this space is and how it's really impacting public health at large.
Awesome. Kevin, thank you so much for taking time out of your day to talk with us. Really interesting topic, and it's been a pleasure and fun time recording with you.
Thank you, Molly. I appreciate it. Again, thanks to your team for reaching out and coordinating this. It's really a great podcast to be a part of.
Have a great weekend.
Yeah, you too. Thanks.
Thanks for listening to Data Unchained, powered by Hammerspace. To learn more, visit hammerspace.com. If you have a guest you would like to hear on the show, email me at molly@hammerspace.com.